Hi everyone, so today's video is going to take the next step with DNA chemistry and introduce how DNA can kind of break the rules that you've been taught. DNA is a much more dynamic molecule than we envision it in this helix, and that's kind of the point of today's lecture, is showing you how far organic chemistry has actually taught us DNA can actually go. So this just is a cool representation of some of the early ideas of the, as it's quoted here, the heritable material. Because remember, people didn't really know what held genetics. They assumed it was maybe it was just like proteins passed, things like that. No, it was DNA. And imagine like just the insanity that comes out that it's this intricate helix with like matching nucleotide pairs that is actually, carrying not just founded in chemistry, but information as chemistry. So even to this day, I still think that it's pretty awesome. And, even to this day, we're still figuring out a ton about it. So this is kind of going to dive into how DNA is a very cool molecule. It's not just this boring static thing. Now, compared to RNA, it is less exciting, but still. So these are what we're working with. As we've covered before, these are the basic structures of purines and pyrimidines. And again, you have your uracil that replaces thymine in RNA. So each of these has some dynamic properties. Okay, we're going to see that they are not always going to exist in these exact conformations. That is going to give them flexibility. And that's the key of today's lecture, basically, is DNA does have flexibility. The key implication of that is that the structures that we're going to see DNA form that go beyond the double helix are very important for that decision to turn genes on and off. And ruining these structures, or maybe causing them to show up too much, can change that dynamic of on and off for genes, and it can change the dosage level of genes. I kind of forgot to make this example, but a good allegory to epigenetics is a light switch, right? It can be on or off, but it can be one of those ones with a dial that you turn it on or on higher or on lower. So again, these are the kind of large scale looks at DNA versus RNA. Again, we'll get into RNA once you get to chapter 6 in that lecture. It is a much more dynamic molecule because these are going to be more reactive. This is a single strand, so it can form like big 3D shapes like we saw with tRNAs, similar to how like amino acids can do this, versus DNA is more than likely always going to showcase this double-stranded structure. And that's very safe and that's very good. But one thing you can start seeing is that this sugar backbone, basically, it has some flexibility here. And I mean, just like in orgo, or like you've seen in orgo, these OHs can attack and form, these specific structures so that these nucleotides can exist in this, the kind of this preferred conformation, but know that they can exist either way. Okay. So keep in mind some of your organic chemistry again, that these bases can determine the 3D structure based on how they are conforming. For example, which carbon in this case is predominating in the endo conformation. In this case, C3 is going to predominate in RNA, and C2 is going to predominate in DNA. This conformation specifically actually makes RNA and grants it a little more dynamic properties. But the fact is, in DNA, we will see where this conformation is going to actually augment the dynamic ability of DNA to move into strange new shapes. Okay? And this is the slide that kind of hits on that key point that I talked about. You do have a lot of flexibility with how these look. So each of these bases, so just like the sugar structure, each of these bases can actually exist in different forms depending on the environment. This is very important because when you think about it, think about all the times where we're like, well, certain genes activate in specific environments, right? So think about the environment of an intestinal cell versus, a heart cell. The intestine cell is, all in this a much like hotter environment. I think at least it's more acidic. Well, I should probably check my physiology before I try to make specific claims. But either way, these are different environments for these cells. So different genes, their DNA will actually maybe be a little different. These structures are going to allow DNA to open and close at certain regions because, as we'll see, you will have certain flexibilities with these conformations that you don't when you just consider these normal ones right here. Okay, So the bases themselves can kind of change. All right, so we've seen this before. This is your standard setup for DNA, okay? Everything's nice. Most of these are nice and flat and planar, and that's usually how we visualize this in these books. And for the most part, you are going to get, this nice, double and then triple down here hydrogen bond structure, and everything's going to be fine. The hydrophobic stacking works. All the chemistry that we've kind of covered is going to dictate how these go. So you can keep that kind of in mind as we go. And I wanted to touch on one way we actually take advantage of how uniform nucleotides are. So HIV is important, and the reason this is in red is that I do want you to know the HIV life cycle because it has been the major teacher for us as far as genomics go. Considering that it undergoes something very special, given that its genetic material in here in yellow is actually RNA. So on its surface, HIV is going to bind to that CD4 receptor on T cells. This is going to cause fusion and integration. This is RNA right here. And like we'll cover in the RNA chapter, RNA was kind of a forgotten molecule for a long time until HIV showed up, because what happens is that this reverse transcriptase right here in green, this is actually an enzyme that is going to turn RNA in yellow into DNA, and that's marked red here. And then orange right here, this protein, and we see it back up here, was packed in with the virus. This is integrase. Integrase is going to take the DNA that you just made from RNA right here, and it's going to pop it into and integrate it into your genome. This blue right here is your DNA. Now it has foreign DNA in it. This red foreign DNA is now going to replicate and also not only replicate this DNA a few times around, it's going to make the products all necessary, including the new RNA genome. So it's just going to make RNA just like normal now, the new RNA genome and all of the proteins necessary to assemble a new virion. And this buds off, and we are off to the races one more time. Okay? So when scientists figured this out, this was a very alien, strange process. We had nothing to combat this until the first drug that we came up with, something called AZT. So remember what I talked about synthetic lethality. What makes and gives HIV its strength? It's this step right here, this reverse transcription, okay? The ability to turn RNA back into DNA and integrate it. So one thing causes this strength to have a weakness is that it needs thymines. It needs to have a rich amount of thymine to replace the uracil that we're going from yellow into red here. So we're turning RNA into DNA. That means we have to convert, in this enzyme, all of our uracils into thymines when we're recopying this into DNA, okay? What we did is we came up with something called AZT. What AZT is a synthetic nucleotide that mimics thymine. It is a fake thymine, okay? What happens is that when you give this to an HIV patient is that the AZT will go in and the viral DNA polymerase is going to gobble it up as though it's thymine, and it's going to incorporate it into the building and budding DNA strand that it's building from its RNA. It needs to do this step to insert DNA into the host genome. The cool thing about AZT is that it's a stalling molecule. It does not have an OH right here that is going to allow any more DNA to be made. It will stall the process and it will inhibit the transcription or the reverse transcription of HIV into DNA. Because again, it's got this little stopper group and it's got no like hooks up here for the next nucleotide, the next actual fresh nucleotide to come. So it stops it in its tracks. Now, this was a monumental find. This turned our survival curves for HIV. They turned them from, something that looked like this, and it's a modest difference, to something that more looked like this. Okay? So now AZT is part of the cocktails that we give to inhibit the virus at different steps of its life cycle and basically hamstring it so it can't evolve out of one step that we're limiting it to. But this trial right here was one of the first ones where we did have to give, for example, placebo and AZT, and this led to a lot of, ethical discussions on, can we give people placebo when they are suffering, for example? But we did need to scientifically know, is this something that can actually work? The issue that we couldn't use, the reason that we had to use human trials in this case was that HIV is a human disease. SIV, simian immunodeficiency virus, is for primates, and that was, what it evolved from. But humans are the host of this virus. We are the only, specific host. You can humanize some animals. We know how to do that now. But to test HIV stuff, you had to test it in the HIV hosts. And that's where this, caused a lot of stir, as it should have. And even today, we kind of have taken lessons from this process, and we much more often use historical data to reflect the placebo than we do ever use like live patients anymore, which is good. Okay. So we talked a little bit about, what AZT is and what that practice is very good for looking at how the nucleotide itself can be useful for other things, right? It can be different for other things. Even our body uses these structures in a lot of cases to form different things, like NAD or NADP+, for example. Like even plants use this. See how you do have this base right here, and it's part of this large, like basically structure, NAD, that we use in our electron transport chain. Even ATP itself, as you can tell, is the precursor of a nucleotide, of adenosine. Isn't that awesome? And then there's GTP too, and that's guanine. So it's really cool how we can use, how our bodies have come to use these structures very often. As you can tell too, all these nucleotides that I've been talking about have double OHs, more reactivity for more dynamic molecules than DNA. So keep that in mind too. So you can use these key structures, these key structures are super important, and they still utilize the basic nucleotide backbone. And so just like AZT, how we synthetically did it, your body's been using these structures a long time because they are really good uniform structures for building or assisting or facilitating other molecular, other molecular reactions. Okay. Now let's get to the part of the class that we'll get, for the first time, I would say, we are going to get into some really alien territory that we've never seen. This is, at least for me, this is like, when I was a student, this is the first time that I saw something like this. And it was a little mind-blowing, because this is actually something that I wish we did better on in biology is, for a lot of the intro level or genetics level, it's like, hey, here's all this knowledge, take it all in type of stuff, and then we expect you to build on it. And that's wrong. That's probably the wrong way to teach such a dynamic field that's always evolving. We need you to build on it, but we also need you to be able to break it down and rebuild it in your own image. And that this material kind of takes you to do that, because it is very different than what you've seen in the past. So the good thing is that with this class, you probably have a very good handle on chemistry. So something like this, and this is a regular DNA, is not going to scare you, right? We have the nucleotide bases, we have the backbone, the double helix winding. What you can start seeing is that there are certain small structures, like major grooves and minor grooves, that form. And the basis of the helix is formed of these major and minor grooves, okay? You have specific turns, you have different angles sometimes. See how the angle right here is changing? How sometimes it's flat, but it can kind of cycle back and forth. This is the beginning of looking at a normal DNA molecule and knowing that, okay, it's not just two lines that are straight up linked all the time, okay? Two other things to keep in mind before we see these three wild structures to take a look at. Take a look at the guanine here in green. See how some of these twists right here in the syn versus the anti position are going to change, in all likelihood, the positions that we see here and the angles that we see them at, right? Thymine has the same capability, although the syn position is very rarely observed. Okay. This flexibility here is why you have so much it's why you have so much flexibility. It's why you can create these strange structures that we're about to see. All right, so some of these are your typical formations of DNA, okay? B, A, and Z. These differ on a lot of different, basically, characteristics. You can see in some of these, for example, so I'll just try and do this in layman's terms first. What I can tell you right now is that B is your typical normal DNA. Let's call it normal, okay? See how it's a nice even curvature. Look at how nice and even these nucleotides are, for example. They're shifting up and down once in a while, but they're pretty much a good, nice angle right here. And it's nice and even. Everything seems fine, right? Okay. Notice A. A is the tightest. See how A is actually shorter than B? A is the tightest, most compact DNA. Look at how, for example, it's so compact that a lot of these nucleotides are at really sharp angles to make contact. Look how some of these nucleotides are even like bursting to the outside just to make their contacts. See how they're kind of like whipping outside into the hydrophobic region? That's because it's so compacted, okay? It's so much tighter packed. And then we come to zDNA. This is very hard to visualize, but zDNA, if you can tell, look at the red strand and how it's winding in this case clockwise, and how these B and A are actually winding counterclockwise. It's because zDNA is actually conformed to go essentially, I don't want to say backwards, because the thing about zDNA is it's the most open. Look how much farther, how much longer the same number of nucleotides in zDNA is. It's much more spread out. You can also tell that a few of these nucleotides are kind of offset in like strange conformations. Most of them are, pretty safely set, but just look at how big these grooves are. And so the first question I'd ask is, if you wanted RNA polymerase to come in and open up one of these genes to start transcribing, is it going to be easier to open up and access genes that are in aDNA, where they're really compact and tight, or are they going to be easier in zDNA, where they are wide open? Okay? It's going to be easier in Z. Transcription factors or other things that come in and open the DNA, they have a lot more access in a big groove like this than they do in a tiny groove like this, right? Okay. I am not going to test you on anything specific. I want the major differences between these. Now, if the specifics help you though, and you're more of a numbers-oriented person and you want the details, this is perfect to go to, and I'd recommend the book or some open materials for this. But you can tell, just like kind of how we went over everything, how the length, for example, I want to see length, sorry, rise, pitch. Yeah, so rise, for example, like how long each of these little segments is going to be. They're much bigger in that zDNA because it's more open. And look right here, the helix twist, positive, but this is a negative twist, which means it's actually running not the opposite direction, but at least the strands are running in different clockwise conformations, which is super cool. And we'll meet the enzymes that actually make zDNA later. Okay. This is probably, I'd say this is the most important slide for these three, just because it's going to showcase different, capabilities. It goes over how, we already talked about B is the most stable. This is your, this is your normal DNA, right? It's nice and clean. You're not putting energy in to compact it the same way you are A, nor is it just like loose all over the place like Z. Another cool difference between B and A DNA is the shift in the pucker here. So remember, A DNA is really short. It's because these two phosphate backbones are now interacting a lot closer than it is in the more in the more open B DNA. And this pucker right here is the reason. Because if you're at C2 and you're doing the pucker, you're going to be a lot farther away than if you were at C3 doing the pucker formation, because now you have moved up almost like 20% or so. But again, most important conformation here is the strangest one, is this zDNA. It's the most open, and it's found near all the promoters for your genes. The other cool thing about zDNA, and what we're studying here, is called topology. This is basically structure of DNA. That's the study of the structure of DNA. The topology of Z favors promoters. It favors regions where proteins are going to bind it and open up that DNA and turn on the nearby genes. So that's why Z is so important. Regions like A, these are going to be in regions of genes that you want closed. Okay, and that's why I want to bring up promoter sequences. Promoter sequences for genes that are on are typically going to be Z. Oh, goodness. Can't draw Z. And that's backwards. But it let me draw the backwards Z, but not the real one. There we go. Promoters that are on, and you are going to transcribe, this area is going to be in the Z conformation. It's going to be open. These boxes need to be accessible. One way to promote accessibility of a gene is to be in the Z conformation. If you want these closed, and if this gene is closed for business in a specific cell, because remember, you need to decide to switch on or off specific cells, you are most likely going to see DNA that is in the A conformation, perhaps. And think of the large like strands of DNA that are in the A conformation, DNA that, is never going to be turned on in a specific cell type. It's just locked in the A position indefinitely. And we have maintenance enzymes, topology enzymes that will keep that structure going that way, okay? So that's it for these three right here. Like I said, it's really cool to see the details that make them, but right here is like these are the big reasons why these structures matter. Open, closed for business. And like I said, the class is driven on epigenetics, and these are epigenetic determinants. Okay, this is where it gets super interesting, and like I said, a little more truly alien. What you're looking at in A, these two conformations, these are possibilities. When DNA lines up in the right way, you can have triplex DNA. What I mean by triplex is nucleotides are conformed in certain ways so that this reaction is possible. This reaction is possible as well. Here you can see the G and the C having the normal 3 hydrogen bonds here, but in the right conformation, and you'll see what I mean by this, if DNA is nearby and it has a C nucleotide right here, in the right conformation, it can be double bonded like this, and you can have 3 bonds. Another interesting structure is B here, poly A purine strand. Sometimes these strands can, and if these are poly T pyrimidine strands, these poly T's and A's can all link up in a big triangle. See how each of these is going to link up right here? Oh, I didn't want that. So sometimes you encounter regions like this where strands of DNA are going to escape and come into contact with other double-stranded helixes and form triple helixes, okay? So keep that in mind, because we're going to go over in the details of how this happens and what it means. And lastly, a megastructure, guanosine tetraplex. In the right conformation, and we'll see what that is, you can have 4 strands basically going like this in large, basically in a close conformation. And if you have G's at all the right heights of this big box right here, they will bind up in this strange but like very cool tetraplex structure. And they actually need a special potassium ion to hold them together. And potassium's the ion right here because it's actually big enough and its field is big enough to stabilize this interaction and keep DNA that, although, it's totally fine DNA, it's not like it's breaking or anything. If it's in this structure right here, and if the composition of the G's are all lined up here, 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 and here, for example, and then down here and down here is same, you can form these like amazing insulated structures. So we'll see what these do too. All right, so let's tackle A and B first. This is a mirror repeat, okay? This is how you can achieve a triplex. What that means is triple strand DNA. A mirror repeat, something like this, where you have a mirrored repeat. Notice how this sequence right here is then the mirror opposite over here. What can happen in this case is that under, some conditions, this DNA helix will just go on being itself because, these are good interactions right here. But what really happens is that all of these T's right here, for example, let's start with these. These T's are binding these 2 A's, but under the right conditions, these 2 T's are going to bind these 2 A's as well, right? This AG right here is going to bind this TC, and this whole strand is going to flip over so that this segment right here is going to have 3 strands binding itself. These 2 original strands, and then this strand is going to flip over here and double bind these. And you're going to have these big triplexes right here. See? That's where this comes from. And this is what you get. You ultimately, so say like, okay, so we're double-stranded right here, and we're double-stranded right here. So back to normal, back to normal. But in the region of the triplex, you have the triplex itself. So you have 3 strands interacting instead of just the 2. But the cool thing that this does is it leaves a single strand of DNA open wide for all kinds of things to come hook onto it. These could be epigenetic activators, repressors, anything it takes to modulate the function of the gene nearby. So that's the key of the triplex, is to not really make the triplex itself. There are some structural things that this can attract, like proteins that'll turn on genes and stuff, but it's to get a nice region of open DNA like this. It's to get this region out and exposed for whatever needs to come and bind it, for whatever function it needs. Could be turning on a gene, could be turning one off, doesn't matter. But the chemistry is simple enough that when you see this structure, it is possible to form something crazy like this. Sorry for the blurry image. Okay, something a little more intuitive is palindromic sequences. So if you have single or double-stranded inverted repeats, see how these are inverted, and they're going to be able to bind each other basically, because these two strands right here, highlighted in red, are actually matching pairs all across. So DNA can actually loop up and form little hairpins like this. And then these four right here, they don't need to bind. Actually, chemically, it's fine that they're just out here like this. But these hairpins can form when you have the right combination of T's and A's that are going to be around and next to each other, and G's and C's. So that's pretty cool. The same thing can happen in a double-stranded case. So if you have a double example of this hairpin, you can form what's called a cruciform, which is two hairpins that basically interrupt a DNA strand. So going from this to something like this, or even, something like this. The cool thing about these structures is that this structure is just pretty normal, it's just regular DNA, but this structure can actually attract like big, massive proteins that can come in and serve as transcriptional regulators. So again, these massive 3D structures of DNA are usually like basically lock and keys to proteins that will then attract more proteins that can say, okay, we're going to turn on this gene right here, for example. So the cruciforms and hairpins, dictated by the chemical structure capabilities of these inverted repeats, they can form 3D structures that proteins will actually bind to. So that's why it's really incredible that although this may not be coding DNA, if you ruin one of these and you ruin what was a good binding pair right here, for example, then you ruin the hairpin and you may lose the function of a gene that this was attracting proteins to that were going to turn on the gene. So it is a lot. And like I said, this is the first time that you're seeing some of these alien structures. So definitely slow me down, ask me questions when you get to this part and, something isn't intuitive. Trust me, the first time that I ever encountered this, I was just like, that's impossible. No way. But you can watch some videos on your own if you want, and hopefully, I can walk you through this too, but it's still really cool. Okay, and this is our final one. We're almost done. The G quartet. This is where you have a strand of DNA, and if it has G's at the right places, and it doesn't matter what's up here really, if you have G's in this planar formation right here, they can actually loop up and down like this and form this big quadruplex thing. So that's why I kept calling it quadruplex. The quadruplex is the massive structure itself. Each one of these is a quartet. So see how the G's can all interact chemically like this and be facilitated nicely? They can do that and form really big ones or really small ones. So that's kind of the cool thing about these, is that they can form like a really big or a really small divot, or they can form like a big massive divot that can like really be used as like a big like structural change in the DNA, that it's like, okay, it used to be just flat right here, but these divots are forming when like certain conditions are right, and this big giant quadruplex can form. So again, it's basically just whenever this interaction is facilitated, you can form this crazy structure basically. This one has some pretty like a little bit better defined roles. So in this case, remember your DNA on the end of your telomeres. So remember from, genetic cell biology, the ends of your DNA in eukaryotic replication, they actually get like chewed on basically, because there's nothing protecting them on the ends, unfortunately. So that's why the ends of your DNA are actually made up of these big G, or the composition of G's that are going to allow for these big quadruplexes, is that nucleases that usually chew up DNA or other nucleic acids, they're not going to be able to act on these structures, because this DNA is in such a strange conformation, enzymes that usually chew on nucleic acids can't come and get them, basically. The other cool thing is they're associated with the nuclear scaffold during telomere, or sorry, during cell division, for example. Sorry about that. And they can be used as structures to attach proteins to that attach to, centromeres or, the plasma membrane or something. So they can serve as like signaling structures. And the other cool thing is that they don't allow, in some cases, telomerase to come and replicate the ends. So remember how telomerase only should be acting on your haploid sex cells, because otherwise it can grant your DNA too long of a life. And you do want your cells to be renewing themselves naturally. You don't want cells to last too long, because that's how you get cancer. That's kind of an essential issue. So again, these G quadruplexes can serve as docking sites for ligands or proteins or anything of the sort that can come and say like, okay, like we're going to block telomerase, or we're going to block other stuff, or maybe we are going to go here or something. Because the word ligand is pretty, it's pretty universal for all kinds of different things that could have different roles. So something so strange like this can turn DNA from your typical helix structure into something that looks nothing like DNA anymore. Thus, things that usually are a problem for DNA aren't a problem for the G quadruplex, and it can also attract like very different specific stuff. So hopefully this wasn't too alien. I actually was super intimidated the first time I saw this, but now to me this seems like one of the more beautiful, amazing kind of introductions into DNA chemistry for me as a biologist. This is where things get really cool for me on the chemistry side. So I really love this stuff. Hopefully you don't need to rewatch it or anything. I know that sometimes it might be easier to watch. I think there's a couple good videos on this, but to be honest, nobody's made a lot of 3D videos on this kind of stuff, because now we're kind of into the area of molecular biology where there aren't as many free resources. Khan Academy starts kind of running dry at this point, and that's why hopefully some of these videos are going to be really helpful. So awesome. Thanks for watching.